artificial chromosome that has all the genes of the original chromosome. So, I've been racking my brain, not too hard, but so it's an artificial chromosome kind of. And that is, think about all the things that a chromosome is going to have to do for a yeast cell. Now, this first one was constructed at Johns Hopkins. They actually used a battery of undergraduates to synthesize, well, this field of science and creating completely from de novo components some biological entity is called I'm trying to find the article before I ran it out here, but my science is in our mailbox. It's synthetic biology. So they've been able to synthesize on machines entire chromosomes for a bacterium, and then put that into a bacterial cell and actually have the cell survive and live and divide and do all the things bacterial cells do. So now they're trying to do that for a eukaryote. So what's this chromosome going to need? It's going to need to be able to replicate. It's going to be able to direct life in that cell. All the genes that are present on the original chromosome have to be on this artificial chromosome. Okay? We've had artificial chromosomes for 30 years. But when we said it was an artificial chromosome, they were extremely small. It's just something we could use to propagate DNA we wanted to propagate inside of yeast that had all its own genes. So this replaces one of the chromosomes in the yeast cell function. Other groups are working on the other 15 chromosomes in the yeast. And then, while riding home last night, I heard an article on NPR about the 20th anniversary of the discovery of BRCA1. So this one I'll post on the library so that you can listen to it. And it's really interesting. The woman King, she actually mapped BRCA1 over 17 years of work to just map the locus, and then it took 4 more years and as a competition to actually clone the gene. So this BRCA1 is a necessary gene for all of us. It's essential. But mutations in BRCA1 are associated with some of the most frequent and severe familial forms of breast cancer and ovarian cancer. So it was a really interesting interview with her on the 20th anniversary of the discovery of BRCA1. And also some of her views on science, some of her opinions on why she was able to do what she was able to do. So, back to our notes. So we ended our discussion going over PCR. And also, I mentioned this last enzyme, PF2, and another one of these extremal phils, these organisms that live in these extreme environments. And so again, it's clever people taking advantage of some basic biology and putting it to good work in laboratories so that science gets advanced. And it also teaches us something about how these genes can function, these proteins, I should say gene products, can function in these extreme environments. So let's give you a practice problem. So on an exam, if I were to ask a question such as this, identify, so ID the potential substrate for DNA pole 3 and draw in final product. And I gave you a choice of very simple structure. So this would be one strand of DNA on the bottom. Let's say it's 5' to 3', an OH here. And then for this one, we go 5' to 3', then OH here. And assuming the presence of DN, TPs, and cofactors, where if I drew a very similar looking structure, but I switch my 5' and 3', or if I drew a third structure, such as that, what will you answer? So, let's see if you paid attention to the last page. So, number one, the question is, identify which of these three would be a substrate for DNA pole, did I say 3 or 1? DNA pole 3, assuming that in the test tubes, you've added all the necessary TNCPs and any cofactors. Things like magnesium and stuff like that have to be in there too. So, A, is this a potential substrate for DNA pole 3? Yes or no? I'm going to say yes. What do I mean a substrate? Can DNA pole 3 recognize this and turn this into double-stranded DNA? Yes? No? Nobody's going to raise their hand. Okay, so what's it take to extend or to synthesize a polymer of DNA? You need a template. So here we have what we call a 3' overhang, right? It's double-stranded here, but we have a piece of polymer with no matching complementary strand over here. So could this be templated? Sure, why not? Now, how does a DNA polymerase act? What direction will it synthesize, and what direction does it read the template? So it reads the template in which direction? It reads the template in 5' to 3'. That's the first bit. Is that correct? How many say yes? How many say no? A lot aren't raising their hands. So what's the direction of synthesis of nucleic acids? 5' to 3'. So if it's reading 5' to 3', or synthesizing 5 to 3, what direction is it reading the template?
3 to 5.
3 to 5. So here we have a piece of DNA that's not double-stranded, so that can be template. Here we have a 5' end. Can this be extended in this direction?
No.
So what direction would that be going in? It would be reading this side 5 to 3 and extending this 3 to 5. Is that going to work?
No.
Yes or no?
No.
No, it's not going to work. Let's look at B. Here we have a 3 to 5' strand. So this is a 5' overhang. This is double-stranded DNA, 5 to 3, with a 3' hydroxyl group. So what does DNA polymerase require in order to synthesize the addition of a base? Of a nucleotide, I should say. It requires a 3' hydroxyl. So does this look like an appropriate substrate?
Yes.
It can read this 3 to 5, and it can extend this 5 to 3. So this would be yes. And what would you draw? You would draw in so we would draw in, let's see, and draw in the final product. Yeah. So we would extend this, and we do 3' hydroxyl group, and this would all be new double-stranded DNA. All right? C. So C looks like this before I drew that in, except that now we have a second polymer, or I should say a third polymer, attached downstream of this first polymer, right? So we have the little primer annealed to this strand, and we have a second piece of DNA annealed to that same strand, and what we have present is a nick. So we have a 3' hydroxyl group. On this side, I didn't draw on the phosphate. There'd be a phosphate group. There's no base without a complementary base. Okay, as I draw these ladders, or rungs on the ladder. So, is this a substrate for DNA pole 3? We have a template. We have a primer with a 3' hydroxyl group. Can DNA pole 3 extend that? Yes or no? How many say yes? How many say no? Why no? Any of you? Somebody help us out here. What if we don't have ligase? The question is, can DNA pole 3 extend this? So I don't care about ligase right now. Yes?
Does it need to have the phosphate group on the other side to attach to it? Because initially the ligase will...
We're not talking about ligase. Wrong answer. What about DNA pole 3 though? The question is pole 3. The answer is no. So why not? What does it lack?
3 to 5 exon nucleus.
It lacks the 3', the 5, but no, the 3' it has, but it lacks which?
5 to 3.
It lacks the 5' to 3' exonuclease activity to remove this strand. So now, which polymerase has that activity? Pole 1. Okay? So this would be similar to the nick that's left after DNA pole 3 has replicated a lagging strand. On the lagging strand, DNA pole 1 would recognize the nick. It would extend that from this 3' hydroxyl, and it would remove this one base at a time using its 5' to 3' exonuclease activity. Correct? I love that question. So now you think you could answer that, right? On the exam. So think about all the things I could change. I could give you Taq polymerase. I could give you DNA pole 1. I could give you DNA pole 3. I could have a mismatched base pair at the end where that's 3' hydroxyl group. I could give you single-stranded DNA. I could give you DNA with a hairpin on the end. Okay? So all of these flavors of question could be asked from the same basic stencil. Yes, Mike?
What if you put primers, RNA primers?
So if I draw a D, so here's my template. So if I do something like this, like that. These would be all single-stranded regions. What would happen now? Would this be a substrate for DNA pole 3? You're shaking your head yes. Why? Wow. If it works on B, why wouldn't it work on D? But now I have a primer in the middle as well, or an intervening sequence. So what would the result be? What would the final product look like in this case? Could DNA pole 3 extend off this hydroxyl?
Yes.
Okay, so let's extend. F base, F base, F base, F base, A type. What does it do now? It stops. Good answer. First answer. Second, streets of Los Angeles. So then what's left here? I think. What about this one? Could it do it again? Lo and behold, yes it can. And what's remaining out of this end? I think. Fantastic. Simple, right? It's like building puzzles. Okay. Eukaryotes. I just love how this is so mechanistic. It's like tinker toys at a very, very, very small scale. Erector sets, Legos, you know. I didn't really play with tinker toys, I'm not that old. Okay, eukaryotes. The replosome. So you have the ribosome, you have the replosome. In eukaryotes, the source that I looked at said there were 27 components that have been identified that are required for eukaryotic replication of the genome. It all sounds the same. Initiation. Initiation in eukaryotes starts at an autonomous replicating sequence. Again, these were identified based upon consensus sequence. They were first identified in eukaryotes in yeast in making the early artificial chromosomes. And how did they know they had a functional artificial chromosome? Well, they basically chopped up the yeast genome and started inserting little pieces into a big plasmid and asked which one can actually be used to replicate that plasmid. And it turned out that there was consensus sequence in all those bits that could be replicated, hence the name autonomously replicating sequence. And ARS. In yeast, there's about 10 of these per chromosome. There's hundreds per chromosome in mammals. And there's thousands per chromosome in rapidly dividing cells of the embryo. So the organism I work with, zebrafish. Zebrafish goes through 10 cell cycles in 4 hours. So it has to replicate its DNA every 4 hours, or it has to replicate it 10 times in 4 hours. The average length of a cell cycle for a eukaryote is somewhere between 12 hours to 24 hours. So this is crunched down and gotten rid of most of G1 and G2, and it's just S. But it's operating 10 times faster in S than a normal cell. And that's because it's using other sites as ARSs. We do not know what those other sites are. And we're not quite sure we have the right ones for the normal divisions. Okay? So what happens during this initiation process? Licensing of the ARS. So what's licensing? When you're going through cell division, you only want that chromosome replicated once, or each chromosome replicated once, before you move on to S phase. You don't want to have multiple rounds of replication. Some organisms do that when they need to amplify the genome. Flies do that in their polytene chromosome, cell division. They need to make extra copies of DNA. So that's called an endocycle, because it just goes from S to G2 to G1. It skips mitosis. Okay. But still, these licensing factors, or there must be licensing, and so there are factors called licensing factors that bind to the ARS. Let me put up here, insert sequence. What they're binding to are thought to be regions that are 100 to 120 base pairs in length. They're AT rich. Licensing factors bind in G1. And then they're degraded, the licensing factors, after replication. So these are synthesized in G1, they bind to the targets in the chromosome. That's one of the properties or one of the functions that's necessary for actually initiating synthesis, S-phase. They're degraded after synthesis, so there's no more replication. So after that region of the chromosomes replicated, these are then broken down. So there's no licensing again until after going through G2 and getting back into G1. So, other issues with eukaryotes. We now have to deal with chromatin. So up to this time, we've been talking about DNA. But in cells, the genome actually exists as a complex of DNA and protein. You know that. %50 of the protein are these histone proteins. And the histones plus the DNA form what are called nucleosomes. A nucleosome is an octamer of histone proteins. Wrapped by 200 base pairs of DNA. The core of this DNA is only 143 to 147 bases. So, histones are an octamer. So how many subunits is that? 8. So if we draw a couple histones, I'm just going to draw them as a barrel. And if we divide this barrel into 8 subunits, let's draw another barrel. And then the DNA would wrap around the barrel. So this would be the DNA, and this would be the octamer. So this whole unit is a nucleosome. So the question is, how do we get a complex of 27 proteins to synthesize this DNA that's wrapped around this core group of 8 proteins? Now we can physically the laboratory, digest the DNA with the nuclease, and that will free the histone, octamer, and its DNA. Okay. So this would be an individual nucleosome. So in short digestion, we see 200 bases. Why? Because with short digestion, the nucleases will cut at the most accessible sites. With long digestion, we get down to a smaller region because then the nucleases trim back the edges. Now, this amount of DNA is significant because in certain types of cell death, nucleases attack the DNA and they cleave it at the sites in between nucleosomes. That cell is not going to recover. It's going to die. But this is a form of death called programmed cell death. Okay. Programmed cell death, also called apoptosis, which I believe I mentioned earlier. So programmed cell death, nucleases attack the DNA. So this is something that is programmed in the genome when cells need to die for one reason or another. Now on the outside of this complex, there's H1. But H1 we can get off with a little salt solution. So it's not part of this core barrel forming structure. So then in 3, we're going to add H1. Not part of the octamer. Remove with, we say salt in the laboratory would almost always mean increasing the concentration of sodium and chloride in the solution. The histones and chromatin. So this arrangement between the histones and DNA is important for packing of the DNA into hetero chromatin versus euchromatin. Heterochromatin DNA is more tightly associated with the histones and other proteins. Euchromatin or true chromatin, the chromatin that accounts for most of the transcribed genes, the histones can actually move a little bit on the DNA. Why would that be important? Well, that DNA has to be accessible to transcription factors, right? And RNA polymerase. What this affects, though, is replication timing. That is, euchromatin, I'm just going to abbreviate it EUK, replicated first. And J. Herbert Taylor, he actually demonstrated this using his tritiated thymidine. So we just didn't work on fava bean seeds. We worked on mammalian cells. And not only did he notice that in the early stages of S, most of the tritiated thymidine was labeling euchromatin, he also demonstrated that really densely packed heterochromatin from female cells. So the bar body replicated late. Okay. So with those same pulse chase experiments, we're looking at semi-conservative, or demonstrating semi-conservative replication. We have histone, so number 6. So this process is that the histones in replication must be removed, or what we refer to as the histones are displaced ahead of the replication fork. So they can't just be pushed downstream because they'll get all bunched up. So they literally, the nucleosome has to be disassembled. Yes? Oh wait, wasn't the last one 5? Oh, I'm sorry. C. Should be a D. Okay, that was my bad. So the histones are displaced ahead of the replication fork. We don't have great data yet for how that takes place, but we know that this is disassembled in the process. Okay. What we know is that after replication, histones added randomly to leading and lagging strands. So I already mentioned that when I was a student, we had this image. So histone on DNA you can actually see in the electron microscope. That tag page. Yeah. Okay, so you can imagine that histones are beads on a string, right? And we know the DNA is synthesized somewhat differently on leading and lagging strands. So the question was, are histones reassembled with old and new histones going to both strands, or in this case, are newly synthesized strands only be deposited on, say, the leading strand or only on the lagging strand? So an interesting thing about histone protein synthesis is histone RNA is only transcribed immediately before S. They do not carry polyadenylated tails, and they're translated immediately before and during S. So it only makes histone proteins a cell when they're needed. Okay? So is there a link between making a leading strand and making new histones, and making the lagging strand and making new histones? And when I was a student, this paper was published. So the electron micrograph, this is DNA, negative stain, higher magnification. You can see those beads on a string, just like drawing this represented there. So these were the data. So in the replication process, if you follow right here, you see beads on one half, and here's a replication fork, and there's no histones on this other. So we were taught that it's somehow inherently built into the system, and then they thought it was probably the leading strand was getting histones, and the lagging strand was only going to get new histones. How do they know there were no new histones? They incubated these cells in cyclohexamide. Cyclohexamide inhibits translation. So there's no new host of histones. It turned out this was an artifact of the experiment. When we have better ways of tagging histones so that we can follow old histones and new histones, it turns out new histones and old histones get mixed as they're going to the leading and lagging strand. Okay? So we can sometimes experiments that look really good based upon the techniques available when somebody revisits that. And they had evidence for reasons why they did this. And they see that this was basically an artifact. It was not representative of what occurs in the cell outside of this manipulation. So let's see, added after replication, and just so you have them in your notes. Transcription and translation. Oops, I'm sorry. Occurs during late G1 and S. And it requires a fracture called CAF1. CAF1 is chromatin assembly factor 1. And this assembles nucleosomes on newly replicated DNA. Now there is an order to the assembly of constructing a nucleosome. 4 parts of the active are added first. I think it's 2s and 3s, and then 4s and 5 subunits are added after that. Okay. So that was D. I'm going to flip it up for E. So that's something that's different between eukaryotes and prokaryotes. But there is additional similarities. In eukaryotes, there's also a clamp. Remember we mentioned the clamp in prokaryotes? In eukaryotes, the protein is called PCNA. This stands for proliferating cell nuclear antigen. So we already mentioned antigens. What's an antigen? Something that elicits an immune response. An antibody was generated that recognized or labeled histology sections of cells during S phase. So this antibody labeled nuclei during S. So hence the name proliferating cell nuclear antigen. So if you're a histologist or a scientist and you want to know which of the cells in this embryo, which of the cells in the intestine, or which of the cells in a cancer were proliferating, you can incubate the slide with an antibody to this, and those that were labeled were the ones that were going through S. We knew that, and we used that antibody for 25 years before we knew what it was recognizing. And it turns out that the target, PCNA, is the clamp for eukaryotes. So recognized by antibody during S phase, localized to the nucleus, and if there's a clamp, then there's another gene, RFC, and this is the clamp loader. So this is similar to in prokaryotes, where there's a clamp loader. What other components are there in prokaryotes and eukaryotes? Primates. Now there's a difference in primates in eukaryotes and prokaryotes. Primates in eukaryotes is part of the DNA polymerase. So it's part of a DNA polymerase complex, and this is DNA pol alpha. Now one nice thing about the nomenclature of the polymerases, we use Roman numerals to identify the prokaryotic polymerases, and we use Greek letters to identify the eukaryotic polymerases. Okay. Should be the other way, maybe, since the Greek Empire succeeded the Roman Empire, but that's another story. Different class. And this also is required for initiation on the leading and lagging strand. So remember, prokaryotes, primates came in, synthesized a primer, fell off, DNA pole comes in, and extends that primer. In this case, it's all one giant complex. But the primase activity is different from the DNA polymerase activity. What this complex does is go through a process that we now refer to as polymerase switching. So in polymerase switching, the primase will synthesize an RNA primer. Now it's within the DNA pole complex. The enzyme will shift its position on the DNA strand or the enzyme complex. And change to DNA polymerase activity. So the DNA polymerase complex will bind to the DNA. It'll synthesize an RNA primer. It'll rotate to put a different enzyme within the complex attached to the DNA. It now will extend and the DNA will be extended off the 3' hydroxyl that's left by the RNA primer. And then this complex falls off. Okay, so the DNA pole alpha stops synthesizing. So now we have a hybrid RNA DNA polymer attached to a DNA polymer on the opposite strand. What's that? It's like D. Yes. Well, yes. Like D. Yes. Yes. So we end up with a DNA strand, but this would be a hybrid, but it doesn't extend to the next primer. It actually falls off very quickly. So we start with 10 to 50 RNA bases, 10 more 50 DNA nucleotides are added, but there's still a long gap before the next deoxyribonucleic acid starts. Okay, so that doesn't run into this. What happens, not only do we have switching within this polymerase complex, that complex is replaced by DNA pole epsilon on the leading strand, and replaced by DNA pole delta on the lagging strand. So even though the primase activity and that initial complex is working on both leading and lagging strand, it gets replaced by two different DNA polymerases, depending on which strands it needs to synthesize. Now, so in mitochondria, DNA fold gamma, encoded by a nuclear gene, will encode or is encoded by a nuclear gene. This replicates mitochondrial DNA. Now, we talked about endosymbiosis. Some additional evidence that mitochondria, and chloroplasts for that matter, were initially some form of endosymbiont, is that this DNA, polymerase gamma, even though encoded by a nuclear gene, is sensitive to certain antimicrobial toxins. Okay? But its toxin sensitivity, this sensitivity changes. And this is one of the really amazing things that I find interesting. Is right now it looks like its sensitivity to microbial toxins is more eukaryotic rather than prokaryotic. So this has changed, one of those other things that has changed from the time I first learned this. I used to say it looked more like a prokaryotic enzyme based upon sensitivity. But as we look at more and more toxins, it looks more like a eukaryotic enzyme. Okay. So, in this case, I don't know. I think it's still kind of out. I have to look at more data correctly. The last few things that we have to do is primer removal. So, primer removal in a prokaryote. 5' to 3' exonuclease activity of DNA to fold 1. Primer removal in a eukaryote. Special enzyme. RNase H. So RNase H is a component. So we have a ribonuclease plus DNA polymerase complex. And what this will do is remove or displace the RNA primer. So how would we know the difference between these two? Between a 5' to 3' exonuclease activity and a 3' to 5, or excuse me, and an RNase H activity. So if we go back to our answer, or our question D. So what if we redrew this and we had DNA fold 1? DNA fold 1, when it got to the next strand, so it could extend. If this is an RNA primer, DNA fold 1 can extend that primer, just like fold 3. When it got to the next RNA primer, it could remove that with 5' to 3' exonuclease activity. DNA fold 1 will actually keep removing DNA as well. But it's not held on by a clamp. So it eventually falls off. But it can remove with its exonuclease activity RNA and DNA. RNase H has a much higher ability to remove the RNA. Okay? Okay, we're going to stop there. On Monday, we'll do telomeres. Hopefully there's no